Regulation of glycolysis and role of fructose 2,6 bisphosphate in glycolysis. The question of this lecture is about how fructose 2,6 bisphosphate regulates the process of glycolysis and how it participates in this regulation. The following is an explanation to understand the hidden parts in regulation. Glycolysis or glucose oxidation as depicted from the figure, starting with glucose, and shown in this figure is a summary of the steps of glycolysis, which starts when glucose is taken up by the GLUT4 receptors, and then when it's inside the cell, it is phosphorylated by the enzyme hexokinase, then converted to fructose 6-phosphate and fructose 1,6 bisphosphate by the enzyme phosphofructokinase 1, which is one of the key regulatory enzymes in regulating glycolysis. Glucose oxidation goes on until pyruvate is formed. We have 2 pyruvate molecules for each 1 mole of glucose. There are 3 important regulatory points, and the most important and rate limiting step of them is the phosphofructokinase 1 reaction, responsible for production of fructose 1,6 bisphosphate. And of course, all of the enzymes involved in glycolysis are increased during the fed state and during glycolysis, as their expression and activation is stimulated by insulin. We have the phosphofructokinase 1, the pyruvate kinase, and the phosphofructokinase 2 in relation to phosphofructokinase So the question now is, what is the fructose 2,6 bisphosphate? Fructose 2,6 bisphosphate is a metabolite formed by the enzyme phosphofructokinase 2 from fructose 6-phosphate during glycolysis. This step is to stimulate phosphofructokinase 1. So the fructose 2,6 bisphosphate stimulates or activates the phosphofructokinase 1 involved in glycolysis, which converts fructose 6-phosphate to fructose 1,6 bisphosphate. This intermediate, the fructose 2,6 bisphosphate, is broken down by the fructose 2,6 bisphosphatase activity of the same enzyme, and this is to inhibit the phosphofructokinase 1. So this enzyme, the phosphofructokinase 2, has a bifunctional activity. It can synthesize and can degrade the fructose 2,6 bisphosphate. So during the fed state, there is increased formation of fructose 6-phosphate, which is a substrate for 2 enzymes, the phosphofructokinase 1 and the phosphofructokinase 2. The first enzyme converts fructose 6-phosphate into fructose 1,6 bisphosphate, and then the process of glycolysis continues, and the other one or second one, which is phosphofructokinase 2, converts fructose 6-phosphate to fructose 2,6-bisphosphate. This intermediate, as we mentioned earlier, serves to stimulate the phosphofructokinase 1 activity of glycolysis. So the phosphofructokinase 2 has fructose 2,6-bisphosphatase activity, and as we mentioned, it is a bifunctional enzyme with a bifunctional activity. So this protein is responsible for the synthesis as well as breakdown of fructose 2,6-bisphosphate. This bifunctional enzyme, the phosphofructokinase 2, is under allosteric control of fructose 6-phosphate. Fructose 6-phosphate stimulates the kinase and inhibits the phosphatase activity of this enzyme. During the fed state, when there is an abundant supply of glucose, the concentration of fructose 6-phosphate increases, and hence fructose 2,6-bisphosphate increases, stimulating glycolysis by activating the phosphofructokinase 1 and inhibiting the fructose 1,6-bisphosphatase. So the sequence of events during the fed state will be when the glucose level increases inside the cell. So the sequence of events that occurs during the fed state when there is increased glucose uptake by cells, glycolysis is activated and there is increased concentration of fructose 6-phosphate, followed by increased production of fructose 2,6-bisphosphate, which stimulates the phosphofructokinase 1 and reduces the activity of fructose 1,6-bisphosphatase, and consequently the whole process of glycolysis is activated. So what about the fasting state? During the fasting state, glucagon is operating and it stimulates the production of cyclic AMP, activating cyclic AMP dependent protein kinase. The kinase activated by glucagon in turn phosphorylates and inactivates the phosphofructokinase 2 and activates fructose 2,6-bisphosphatase. And this will lead to a decrease in fructose 2,6-bisphosphate with inhibition of phosphofructokinase 1 and releasing the inhibitory effect of on fructose 1,6-bisphosphatase, and consequently there is reduction of glycolysis and stimulation of gluconeogenesis. Hence, gluconeogenesis is stimulated by a decrease in the concentration of fructose 2,6-bisphosphate, which inactivates the phosphofructokinase 1 and relieves the inhibition of fructose 1,6-bisphosphatase. So the sequence of events during the fed state will be when the glucose level increases inside the cell. So the sequence of events that occurs during the fed state when there is increased glucose uptake by cells, glycolysis is activated and there is increased concentration of fructose 6 phosphate, followed by increased production of fructose 2,6 bisphosphate, which stimulates the phosphofructokinase 1 and reduces the activity of fructose 1,6 bisphosphatase, and consequently the whole process of glycolysis is activated. So I hope this explanation could explain the role of fructose 2,6 bisphosphate in regulating the process of glycolysis. And you can find some other relevant information about this subject and other topics in this site.